Hello everyone, I am Professor Charu Dogra from University of Delhi Ramjas College and I have been continuing the series on biotechnology. Today I will be talking about the molecular diagnosis of genetic diseases. The particular learning objective of this session will be to understand the molecular diagnosis of diseases, particularly the PCR oligonucleotide ligation assay, which is generally called as the PCR-OLA method. So, we will be discussing about the principle and we will be also discussing about the use of this method for the detection of mutations in sickle cell anemia disease, cystic fibrosis disease and even SARS-CoV-2 mutations. We will talk about that. For educational purposes, the reference used for this apart from the internet sources is Glick and Pasternak and Patton, and particularly for the educational purpose. So, we have been talking about the molecular diagnosis of diseases. Molecular diagnosis is the application of molecular biology tools and techniques to identify specific genetic mutations that are responsible for various hereditary diseases. Now, initially this detection or diagnosis used to be done by various biochemical or morphological assays. But the prenatal diagnosis of Mendelian genetic disorders has been limited to the conditions for which the biochemical defect was known and expressed in the fetal tissues such as the amniotic fluid cells, chorionic villi blood, skin, liver, etc. So, the biochemical assays, the biochemical defect needed to be known and expressed, and that is how the prenatal diagnosis of these Mendelian disorders used to be done. Now, of more than 2000 known Mendelian disorders, less than 100 are amenable to prenatal diagnosis using the biochemical or morphological markers in fetal tissues. So, this was a big limitation because every disease does not manifest at the level of biochemical or morphological markers. Then also there are not any diagnostic tools which can accurately assay these changes and therefore predict the generation of the genetic disorders. So, some molecular methods and tools and techniques were devised so that the various genetic disorders can be detected and diagnosed prenatally. The gold standard method to identify the mutations is to directly sequence the PCR amplicons. Now, we have also discussed about the the evolution of sequencing techniques starting from the Maxim Gilbert to Sanger sequencing and leading to all the next generation sequencing. So, it just comes to mind that why not just sequence the PCR amplicon of the genes associated or the mutations that has been caused to manifest these diseases. The thing is that these technique, the sequencing technique, nevertheless a lot of cost has been reduced on the next generation platforms also, and even in next generation sequencing platforms the time is also less and day by day the evolution is taking place, but still for prenatal diagnosis this method is expensive, laborious and time consuming, particularly if we have to have a large scale application. So, although the gold standard is to sequence the PCR amplicons, but because of its limitations being a little expensive, laborious and time consuming, nevertheless the kind of advancements that has been occurring for the NGS platforms, there had to be devised certain other tools and techniques of molecular diagnosis which can accurately, quickly and with less cost, very cost effectively detect and diagnose the various Mendelian disorders. In our last sessions, we have discussed about the diagnosis methods based on the PCR-RFLP, which is the restriction fragment length polymorphism. So, the gene which was used to be mutated, for example, in sickle cell anemia disease or in the beta globin gene or in cystic fibrosis the CFTR gene, that is amplified or a region of it is amplified and then digested. Now, because of the presence of restriction sites, there is a restriction fragment length polymorphism, and because of the occurrence of the mutation, and if that gene at that particular region contains a restriction site, then it may get altered because of this RFLP. So, with the presence of the mutation, certain restriction sites can be abolished or certain restriction sites can be generated for a particular enzyme as well that can be detected by gel electrophoresis or other methods, and therefore this PCR RFLP can be a good diagnostic method for the detection of diseases. Another method that we discussed is the PCR SSCP method, which was the single strand conformational polymorphism. So again, the gene in concern was PCR amplified, it was melted, the denaturation was done due to the change in temperature or due to the change in salt concentrations or the pH. The double stranded DNA is then denatured, and then it is allowed, certain conditions are given so that the single strands can fold on itself. Because of the presence of the complementary regions, they can fold and give rise to a certain structural conformation. Now, because of the particular mutation, it can be a SNP or insertion or deletion, whatever the case may be, then the conformation would be altered, and this conformation can then be detected on, for example, polyacrylamide gel electrophoresis, neutral polyacrylamide gel electrophoresis, and the differences in the conformations of the wild type and the mutated gene can then be detected, and therefore the diseases can be diagnosed. The last method which we had already discussed is the ASO dot blot method, which is basically the allele specific oligonucleotides. So, allele specific oligonucleotides are synthesized and allowed to hybridize with the gene in concern causing a particular disease, for example, the beta globin gene or the CFTR gene. These ASO, I will just review a little bit, that the allele specific oligonucleotide technique utilizes the synthetic oligonucleotide probes, which are about 19 residues in length, which is complementary to the normal and one to the mutant allele. So, what happens is that under appropriate conditions of hybridization, the oligonucleotides will bind only when there is complete complementarity. This characteristic of the hybridization we had already discussed in various hybridization techniques as well, such as the southern blot hybridization. So, we had talked about the term which is called as the stringency. So, when we alter the conditions, this stringency can be altered. Now, higher stringency or lower stringency basically means that under certain conditions, whether the complementary base pairing is allowed, which is very, very strict. The higher stringency, it is like it has to be 100% accurate, and under lower stringency conditions, it will allow certain nucleotides to be altered and still the hybridization will take place. But obviously, for ASO detection method, the 100% or complete complementarity is the conditions are provided so that there is complete complementarity, and in this case, for example, it is just schematically shown here that in the wild type, if it is a wild type probe, then the wild type allele specific probe is going to hybridize to the single strand gene of interest, the region of interest, but the mutant probe is not going to bind it. For example, there is a mutated nucleotide or there is an altered nucleotide, a SNP, single nucleotide polymorphism in the center of this particular gene. So, the mutant probe is not going to be bind, and then these probes, they can be loaded with various detection molecules. It can be colorimetrically detected molecules, or it can be fluorescent labeled, or it can be radioactively labeled. So, whatever detection method we need to apply, they can be tagged with such molecules, and then they can be allowed to hybridize. So, as we can see, they can be the wild type and the mutant probes can be tagged with different tagging molecules, and then they can be detected to give rise to different signals that can then be detected, or the wild type is tagged with one, and if both of them are tagged with the same, then we can just see that in one case it will bind and the other probe is not going to bind. So, this is allele specific oligonucleotide. Now, why did I review this? Because the method that we are going to discuss today is the oligonucleotide ligation assay, which is the OLA, called as OLA. So, this is again an important method, and in this again we use the allele specific probes. Two oligonucleotide or probes, usually 20 mers in size, are used, which are then hybridized to the DNA target PCR amplicon. So, again we will PCR amplify the DNA target. The DNA target is again the region where the mutation. So, in this case again the mutation needs to be known. It needs to be known that such a kind of mutation is there. For example, we have discussed in beta globin gene where there is a difference in one nucleotide in the 6th codon which changes the 6th codon of the beta globin gene and therefore gets altered. And in also CFTR we have discussed about a lot of mutations, many number of mutations we have discussed, and one of them was, for example, delta F508 and other kind of mutations which are there. So all these kind of mutations which are known, we can design the primers to amplify such regions containing the mutations, and then these DNA target PCR amplicons we can hybridize with 2 oligonucleotide probes. Now these probes, they are hybridizing in juxtaposition to target DNA, are joined by DNA ligase. So what is juxtaposition? Which is basically the proximity, they are one after the other. So one probe is basically the ending at 3 prime and the other probe is starting at the 5 prime. So what we use is generally 3 kinds of probes. One is against the wild type, the second is against the mutated, and the third one is a common probe. So we add these 3 probes and then we see how the binding occurs. And after the binding, because the common probe is going to be bind to the adjacent region of the DNA target that we are looking at, and the DNA target, whether it is wild type or whether it is mutated one of the oligonucleotide probes is going to bind to that. So at one instance, 2 DNA probes will be bound to the target region. One is the wild type or mutated probe and the other is the common probe that we are talking about. Before I show you the picture in which it is more clearly understandable, one thing I would like to discuss is the conditions for the ligation. So the important part is that one thing is that the hybridization of the probes to the target DNA is essential. So when we are discussing the OLA method, the first thing is that the probes need to be bind or hybridized to the target DNA. Now when we synthesize oligonucleotide probes which are 20 mers, it is not necessary, and if we are using, for example, the if we are using the genomic DNA or a plasmid DNA where the target DNA is target region is present, if we are using that to hybridize the probes may bind at various other places. But the probability of the 2 probes, the one which is either the wild type or the mutated probe and the other probe which is immediately downstream to that, the common probe, both binding side by side is very, very less. So they may bind to other regions in the genomic DNA if we are using genomic DNA. If we are using a PCR amplicon, then obviously we are just using the target region and therefore the probes will be very specifically bind to the target region only that we are talking about. But if we are using the genomic DNA as the source material, then the probes may bind at different places in the genomic DNA as well. Again it depends upon the stringency, but if it allows, then it may bind to the certain other regions as well. But both the common probe and the wild type or the mutated probe to bind just adjacent to each other is very, very less. So obviously the second condition for this ligation to occur would be that the probes must lie directly adjacent to each other in a 5 prime to 3 prime orientation, because the next step after hybridization is that we need to ligate the 2 probes, the wild type or the mutated probe and the common probe. So we will use a ligase such as a T4 DNA ligase or any other, even if we are using a thermophilic organism, we can use the thermophilic ligases as well. So we can use the ligase for the 2 probes to be ligated. So these 2 probes must be present in juxtaposition to each other. The proximity is very important. It should be directly adjacent to each other in a 5 prime to 3 prime orientation. And the third condition for the ligation would be that the probes must have a perfect base pair complementarity with the target DNA at the site of their joining. So as I said, because we are talking about the single nucleotide mutations, if it has to be highly specific to detect even the single nucleotide polymorphisms, then what we need is a perfect base pair complementarity. So 100% complementarity should be there so that it can detect even a single nucleotide change that is occurring in the mutated gene. So these are some of the conditions which are required for the ligation. If the hybridization occurs and the conditions are met, then it can be ligated. So looking at this picture, what I was talking about, that what we use is these two different kinds of probes. So one, as you can see, is a wild type probe, and the second you can see is a mutated probe. So this wild type probe is going to bind to the wild type sequence, and the mutated probe is going to bind to the altered sequence where there is a nucleotide change that is present. And then there is a common probe which is going to bind to the lower, just next to where this is going to bind. So this 3' where it is ending, the 5' of this is going to be starting. So as you can see here, this is a wild type target region, and this is a mutated target region. So in the wild type target region, when we allow the conditions for the hybridization to occur, the wild type probe is bound here, and just next to it this common probe is binding. But in the mutated target region, the wild type region is not able to bind completely because of the difference in the one nucleotide where it cannot bind, and the common probe is going to bind as usual. This is the mutated region. So in the mutated region, you can see that the wild type is not going to bind because there is going to be one change in the base pair. But however, the mutated probe is going to bind here. The common probe is going to bind normally because that is everywhere there. Then after this hybridization, the ligation is done. So when the ligation is done, the DNA ligase is going to catalyze the ligation of the two probes which are just in complementarity and in continuation to each other. So in this case, it is going to ligate the common probe and the wild type probe, which is lying just adjacent to each other, the 5' and 3', but not to this one because they are not juxtapositionally positioned. So they are not just next to each other, and therefore the ligase cannot ligate these two products. So you can see in here the wild type probe is going to bind to the common probe and create this ligated product, and in this case the mutated probe is going to bind to the common probe and create this kind of a ligated product. Now both the ligated products are also tagged with, so when the probes are used, they are also tagged with different detection molecules. So it can be a chemical detection, it can be radioactive detection, or it can be any enzyme substrate detection, fluorescence detection, any different kind of detection methods where we will just load a detection tagging signal also here, so that we can just perform the detection mechanism, and therefore the signal can be given. So the two tags can be different, and therefore we can see that the difference in the detection pattern would be there, and we can identify that whether the gene is a wild type gene or whether the gene is a mutated gene. So I hope this principle of PCR-OLA method is clear to you, and again we will take in further cases how it has happened. So when the probes are mismatched at their junction, they are not ligated. Ligation products are then detected, as I said, colorimetrically in, for example, in an ELISA format where enzyme linked immunoassay is done, or by denaturing, such as the polyacrylamide gel electrophoresis. Because when you denature the gel ligation product, then again the conformation would alter, and that conformation can be detected by running on the gel electrophoresis as well, or they can be tagged with different molecules, the chemical molecules, and therefore detected colorimetrically as well. This is one of the method I again show here to make it clearer that how this detection occurs. So these are called as the capture denaturation detection method, which is the CDD method. This is the mostly widely applied method for the PCR-OLA assay. So in PCR-OLA assay, what we do is we start with the PCR amplification of the DNA target gene target, and then we are going to hybridize it with the wild type or the mutated probes, and then you are going to ligate these probes there. Then after the ligation, as you can see here, then they are loaded onto, in this case this is a plate capture method, this is a paper capture method. So basically the support which we are using here is a plate, and in this case what we are using are the paper strips. So the reference of this particular method, the paper reference paper is given underneath this picture. So you can refer this as well for detailed aspect of how this is done. So I will quickly explain it. So what happens here is, for example, these are the probes that we are talking about. Now these probes, the wild type probes or the mutated probes, have been loaded with this different detection here. So you can see either it is A or it is D. So it is a alkaline phosphatase or it is the other molecule which is there. The details of the acronyms are given here. So the different detection is there. Then at the other end of the probe, it is bound to the biotin. So this biotin streptavidin enzyme, it is loaded onto either the plate. So on the plate there will be streptavidin, and this biotin streptavidin complex is going to load these probes. And one important aspect, as you can see here, is that the multiplexing can be done there. So number of probes can be used simultaneously, because it can be loaded onto the same plate or a small strip. Many different probes can be used and the detection can be done. So it is very useful in cases where there are a number of mutations that lead to the diseases. For example, the CFTR gene. So number of mutation causes the alteration in the functioning of the CFTR gene and the manifestation of the cystic fibrosis diseases. So multiple probes can be used and simultaneously detected to see if there is one mutation or there is a compound mutations or there are 2, 3, 4, 5 mutations that are occurring. So what happens here is, the first thing is the capture. So these ligated products are captured onto the plate or onto the paper by this biotin streptavidin complex formation. Then the denaturation occurs. So the denaturation is going to remove all the products which are not ligated. So you can see these products are not ligated here. Similarly, these products are not ligated here. So only the wild type or the mutated probes where the complete ligation product is there will remain. The rest of them, only the single probes which have not been ligated are going to be removed. So denaturation conditions are given and the ligation product remains on the plate or the paper, and the rest of them just goes away. Then after that, the antibodies using for this detection to occur. For example, there is a horseradish peroxidase here, or for there is a alkaline phosphatase thing here. So this is a POD's peroxidase we are talking about, dioxygenin, and this is the alkaline phosphatase which is there. So they are going to be bound to their anti molecules, and you can see that they are basically binding here. In here the same POD is used, but in here two different tags, one which is tagging the wild type and the other which is tagging the mutated probe has been used. And then after this denaturation, the detection is done. So this reduces, for example, in here the mutated one is reducing the whatever the compound to the yellow product that can be detected, and the other one is doing converting this TMB into these are the benzidines chemicals that we are adding. It is converting this into a brown product. So you can detect these two different, you can do the washings for one can be detected at, for example, 405 nanometers and the other can be detected at 450 nanometers. So colorimetrically these two different separate compounds can be detected, and we can see that whether there is a wild type gene that is present or a mutated gene that is present. And in this paper strip method, we have used that tagged both of them with the same probe, and when both of them are basically tagged with the same probe, there is a difference in the occurrence of the bands that we can see. Because the intensity is different, and we can see that these products will be, so there is a background that is occurring, or it is only binding to whatever the mutated probe or whatever, and then it is giving a different band out here. So this is called as the capture, denaturation and detection method. This is the most widely applied method for doing the OLA PCR. So I hope this method is again clear to you. the principle of OLA PCR as well as this detection method, that how the detection is occurring. So, let us look at that how the diseases that we have been talking about. So, I have taken the sickle cell anemia disease, that how it can be diagnosed by using a PCR OLA method, and I have also used the again cystic fibrosis that we have talking about, that how cystic fibrosis mutations can be detected by the PCR OLA method. Additionally, I will also be showing the SARS-CoV-2 detection method that can be, which caused the COVID-19, that how that can be detected by PCR OLA method as well. So, this sickle cell diagnosis, as you can see, two different probes are used. These two different probes, the 131 probe and the 132 probe, one is for the wild type one and the other is for the mutated probe. So, you can see here that there is an alteration of one of the nucleotide in the beta globin gene that is basically causing the change of the amino acid to valine and therefore causing the sickle cell anemia disease, and this is the common probe P133 which we are using here. So, these are the common probe, and these which is just adjacent to the region where this mutation is there. So, it has to be the mutated gene, just next to that this has to be there. So, this is basically where the ligation needs to be occur, the mutation has to be present there. So, these two different probes are there. Now, again I will just review the inheritance of the sickle cell disease. It is a recessive genetic disorder. So, there would be a dominant one where there will be both the alleles are wild type alleles. There will be carriers where one wild type allele and one the mutated allele would be there, and the disease will be manifested in a homozygous recessive condition where both the alleles will be the mutated alleles. So, as you can see, when the products were ligated and when these products, so there are it has been done with the genomic DNA, the plasmid DNA where they were present, and even the total cells, they were just in a one step method they were isolated, and then it was basically used the different B131 and B132 probes, and you can see here this is beta A and this is beta S. So, this is a wild type gene and this is the sickle cell mutated gene which is there, and when the hybridization is done, then that hybridization and detection, this is the hybridization and detection of the ligated products. So, of the ligated products you can see where the wild type allele is present, it is hybridizing with the B131 which is containing the wild type probe. However, where there is a sickle cell mutation where there is present, the signal is present in the B132 probe which is there. It is present in both the cells, the plasmid as well as the genomic DNA, obviously differing in the intensity because of the amount of the concentration that has been taken, but the probe binding is showing and reflecting very clearly whether the beta globin gene, the target region is a wild type region or whether it is a mutated region. The B131 probe, which is against the wild type, is binding wherever there is a wild type gene there is present, and this signal is only shown when there is a sickle cell anemia. So, this can easily, so the blood is drawn from the fetal tissues, and then the genomic DNA, even the cells can be directly used or the genomic DNA can be isolated, and as I said, even the PCR amplification can be done for the targeted region, and then the probes can be used the probes can be used to be hybridized and ligated, and thus the PCR products can then be detected, and it can be ascertained whether if both the alleles are wild type, then it is not carrying a disease. If one allele is wild type and one allele is mutated, that means it is a carrier, and if both the alleles are homozygous recessive, that means it is going to have that sickle cell anemia disease. This is again one of the thing where the PCR amplicon was used, and in the PCR amplicon you can see there was a peak, a 120 base pair peak here present. Now, they have been detected with different target molecules. So, this was a case which was the wild type, and you can see the peak here giving by a signal of one particular detection molecule. The other was tagged with a different molecule, but it is given at the same point, but it was a different color that is given here. So, this is also telling you whether it is a the sickle cell gene or whether it is the wild type gene, and in which the gene was deleted, there was no peak which was present at all. So, this is the result of a PCR-OLA method. Similarly, the cystic fibrosis diagnosis can be there. The cystic fibrosis diagnosis, again, you can see different signal peaks are present in different wavelengths. There is a blue, red and yellow wavelengths that can be seen, and different mutations can be detected by these different peaks. In total, for example, here there are 29 peaks or 30 peaks which are present, but if one of the peaks is missing, then it can point out, like for example shown here, that this F508, which is the most common mutation, is missing. So, if that peak is missing, that means this particular probe is not binding or ligated here, and the mutation has occurred, and therefore it is a cystic fibrosis manifestation can be there. The last I discussed is the SARS-CoV-2 mutation detection. It is again the same method. What we use is the cDNA here, because it is an RNA molecule. So, we use the cDNA molecule and we use the wild type probe, and we use a mutated probe, and along with that we use a common probe. So, when we ligate it together, so the wild type probe and the common probe binds together, but the wild type probe cannot bind when there is a mutation which is there. So, the nasal swabs are taken, the genomic DNA or the cells are taken there, the PCR amplicons can be taken, and then these specific probes are allowed to hybridize to that gene regions and then ligated, and as they ligate, you can then be captured on the plate or paper, as I explained earlier, and then because of the presence of the certain detection molecules, and a lot of multiplexing can be done here. So, this is the NPlex that they have been talking about. Certain specific NPlex probes have been also used, and these probes are carrying the different detection molecules as well. So, there is a biotin by which it can be captured onto the plate or the paper, and then this NPlexing can be done so that simultaneously many samples or simultaneously several mutations can be detected by this PCR-OLA detection method. So, in general, this PCR-OLA detection method is highly accurate. There are many advantages that we talk about here, but the basic limitation is that there can occur some kind of a polymorphism there, and then certain important for the ligation etc., certain instruments are required which can be there, but there are so many advantages that it predicts or detects with high accuracy. And again, certain modifications have been applied so that the detection method is more precise, but again a lot of advantages are there for the PCR-OLA ligation method, and it is a very common method to detect the different genetic disorders. Thank you.